No, but you can't because
She said I can, right? Yes. All right, I asked. All right, so yesterday I presented something that was at the interface between biology and material science on the other end. I found working in this weird domain of nanomedicine that a lot of times engineers that come up with particles don't understand very well the biology of the body. And at the same time, working with cancer biologists, very often they don't know much about the ability of tissue engineers to create materials that can really benefit the study of the biology. So this is the other way around. At this time, what I'm going to present is how we can use materials that we develop for tissue engineering, so scaffolds for musculoskeletal tissue engineering typically, as a way to improve the quality of cancer research that we do. And what I'm going to focus on today, it's the fusion of this material, which is a porous scaffold, and these materials, which is a nested silicon structure like this, embedded inside a polymer capsule. The way we make collagen is pretty special in the lab. We take a great care of assembling in different ways. So we have formulations that are injectable, like pastes, very flexible membrane, thin, and porous scaffolds that can be also mineralized. So the common denominator of these three pictures is collagen. Collagen is at the base of the three formulation. One case you have sugars, one case you have elastin and other proteins, one case you have minerals. But the important thing is that once it's assembled in vivo, the collagen is ultra structured, which means that it maintains the classical D bending 67 nanometer apart, which is key for cell recognition and entrapment of growth factors. We published a number of papers on this technology, so this is now a pretty standard technology in the lab. And the way we combined, as I mentioned before, to focus on the scaffold and on the polymeric particles, the way we merge the two is that we put the particle, which is now in light blue, right under the skin of the collagen scaffold. So we embedded particles, for example, releasing different type of proteins, different types of factors in different layers. And we kind of demonstrated this in this little cartoon here. You have different, kind of a mosaic type of structure. So you can embed 3 different drugs and combine them in different ways. You can combine them in 2 layers. You can have a gradient. The bottom line being that you can control very tightly the release rates in time, but also in space. So it's kind of a spatial and temporal control. And now you see here that all the red spherical particles that I showed you before, they are entrapped in the middle of the blue collagen wall. So this is to guarantee that once you have the final scaffold, which is a monolithic structure, so there is no layers there, it's just one big piece. But when you look at this structure under the fluorescent microscope, what you see is that now you have the boundary between 2 different areas, and these allow you to pattern in space a 3-dimensional scaffold so that it can release different type of drugs. And you see here, this is a higher magnification that every single particle is covered. We then started to use these scaffolds for a lot of tissue engineering applications. And this because the other beauty of this is that once the particles are stuck inside the collagen scaffold, macrophages and other cells of the immune system cannot really remove it. And so you have a consistent release over time. And what I'm going to talk to you about today is how we use these scaffolds for a completely different reason. Not for tissue engineering, but to kind of model cancer in a better way. And obviously, these scaffolds support the cell growth very well. You see it over time, from time 0, when you see just a few cells. Over time, the whole scaffold gets colonized. All the pores get filled with cells. And the 2 cell type that I'm going to talk to you today about are 2 very important breast cancer cells. And these are very pretty much 70% of all the papers you see have these two cell types. These are derived from patients. They're primary breast adenocarcinoma or an invasive ductal carcinoma. So the reason why I picked these two is that I wanted to compare how two cell types, a primary tumor type and a metastatic cell behave. They obviously look very different. These are glomerular, they create these dome shapes. These are more stellate. But once you grow them on the scaffold, what happens is that these cells start to resemble very much the appearance and overall organization that you see in a real xenograft or in a mouse model. So these are the same cells injected in a mouse, and we did histology later on to show it. And the reason is that a few years ago, Hanan and Weinberg published a very important paper in Cell about the hallmarks of cancer. And if you look at them, it's very difficult to recreate these hallmarks using an in vitro 2D culture system on plastic. And the reason is that cells in the body don't grow in 2D, certainly don't grow on plastic, and they can't really recapitulate all the different aspects of this. And what I want you to focus on is on cell energetics, because cells in cancer change their metabolic behavior. Proliferation, obviously. I want you to focus on angiogenesis and invasion and metastasis, because these are the four things that I'm going to describe to you today. So the first thing to notice is that these cells in 2D, they always look the same. When you start growing them on the scaffold in 3D, you start to see the appearance of these giant cells. And we'll touch base later on that. But you see that it increases not only in percentage, you have more of them, but the increase in size is also massive. There is a change in proliferation. Cells in 2D, they just grow until they reach confluency. And there is this typical expansion phase and then a plateau phase. That is not really what happens in vivo when cells have a much slower proliferation phase. And we were able to kind of mimic and recapitulate that type of growth on the 2D scaffolds, on the 3D scaffolds. And we check a number of other things. I mentioned before the giant cells. These are very important because this is something that you find also in vivo, in the xenograft, but we could find also in 3D. And these cells are senescent cells. So they express beta-galactosidase. And the reason why they're important is these are the cells that are very often responsible for resistance to therapy and for recurrence of the cancer. So when you treat cells in 2D, these cells never show up. All the cells get killed by a treatment. You never develop any senescence, any resistance, and so you kind of miss a little bit of the whole story. The second part that I asked you to focus on was the angiogenesis, which is triggered by hypoxic environment. And the hypoxic environment, again, in 2D, if you look at HIF1 alpha, there is no expression in 2D. There's no reason for the cell to express that. But when you start growing in 3D, you start to see all the cells being positive, which is something that happens also in vivo. And when you look and you inject in a mouse an hypoxic probe that just measures where oxygen is not available, you see that this staining co-localizes with the expression of HIF1 alpha. And this happens also in the 3D culture system. I mentioned the other typical feature, which is it's the metabolic switch, which for breast cancer cells, it's also known as Warburg effect, something described like 1918, I think. And what you see is that now you start to see the overexpression of GADPH. First of all, you start to see the glucose transporter that in 2D it's never expressed, in 3D starts to go up. So you see the same features that are characteristic of the metabolic switch in cancer. And then the final one, obviously cancer cells do metastasize, especially when they are in very strong hypoxic conditions. And what we saw here is that this is a scaffold. It's a cross-section of a scaffold. You see in red the collagen, in yellow cells. And you see that at day one, all the cells are kind of clustered also in this case in the middle. But over time, they migrate outward. So they respond to the fact that there is a necrotic core organized that forms in the center. What did I do? And they kind of respond to that by migrating towards the edges of the scaffold where the oxygen tension is instead higher. And you can measure that very carefully, like in terms of numbers, but you can also understand what proteins they start to overexpress. So raw GTPase, it's involved in actin dynamics, in cell movement, and you see that, that the gene goes up all in the 3D. You'd never see it in 2D. And the cells organize themselves in this very typical feature that is called Indian filing. It's like they're like line up one next to the other, and this is because, especially in the case of MDA, there's very aggressive cells. We then try to recapitulate all of the gene expression patterns that occur in 2D and 3D, and you see that there is a complete black and white, or red and green in this case, flip of the expression. And so there are like a number of genes, and these are genes involved, again, with metastases, like metalloproteases, LOX is another one for metastatic spreading, all of the genes for metabolic pathway. And the important thing is that, again, in 2D, you have a completely different situation than in the 3D and in vivo. And so the idea was just to try to get closer to the real situation in an in vitro setting without always using an animal. And what happens when you inject, so you culture cells in 2D all the times before implanting a tumor in a mouse, but when you precondition these cells in the 3D scaffold, what happens is that the tumor takes on much faster, it grows more aggressively, it produces more blood vessels, it actually gets closer to the look of the real cancer. And the final slide, which is probably the most interesting way to use these scaffolds, is to test drugs. We tend to test drugs in 2D, on plastic. Those are not the right cells to treat. Those are not the cells that you will actually find in the real cancer. And you see that because when you treat to the cultures, like for example in this line, at any concentration of drug, this is doxorubicin, cells die. But when you do instead the same experiments in a 3D setting, the cells do not die as quickly, and they actually get closer in terms of treatment to the IC50 that we measured in vivo. So just without trying to say that we were able to perfectly match the complexity of events and the variability and number of different cells that occur in vivo in a real tumor, we were able anyways to kind of get the situation a little closer to what happens in a real cancer using a scaffold that is very simple to make, very reproducible, can be made in a 96 well plate, and it's just my attempt to raise the level of cancer biology from plastic on 2D to something that is a little bit more physiologic. And I want to thank the people that did the work. In particular, this is the work of Silvia Minardi, who did all the collagen work with Franci. John did all the imaging. Juping did the animal work. But in particular, I need to acknowledge the collaboration with this group of cancer biologists that have been the first one to listen, and that we've been working for a few years at this project now, and they are in Italy, in Forlì. Thank you.
Thank you very much for the great presentation. You also, I couldn't see all the genes you tested for microarray analysis, but you also test PGP or other drug-resistant marker efflux pump, or it's just a kind of physical
This
barrier?
Was not to understand that treatment. We didn't do this after treatment. We were doing just this to understand the evolution of the cancer cell in the scaffold. So we were more interested, actually this is just 1 and 3 days time point. We did also later time points. We were just my curiosity was, do the cell evolve in the 3D scaffold in the same way that they change and mutate in the in vivo setting? Because you don't see a lot of change in 2D. They're kind of exposed to the same situation, same environment all the time. But I did not look at the efflux pumps.
Thank you so much.
Yes, thank you, Ennio. If we stay in this slide, you have a huge difference between 3D and in vivo, as much as from 2D to 3D. So do you think that are we having only a collagen scaffold you can so much recapitulate, especially with this type of cells that are very sensitive to extracellular matrix? Adding laminin or stuff like that could be very interesting.
Yes, so that's a very good point. As I said, never I would say that I was able to recapitulate everything, but it's important that at least you simplify the solution. Trying things in 3D in vivo sometimes is very difficult to understand, and so you dissect one thing at a time. Now, for what pertains the collagen matrix, you're absolutely right. What happens in a real tumor is that the elasticity of the extracellular matrix varies greatly. And these two cells, even if we call them always breast cancer, they're completely different. One, it's a real epithelial cell. It sits in the ductal, in the ducts. And the other one, instead, it's invasive. It's a more mesenchymal type of cell. And they deposit a completely different extracellular matrix, which is something we're learning now. So what we're doing now, which is very tricky, we're trying to measure the stiffness of our scaffolds, adding elastin, laminin, and trying to match the elasticity with the elasticity of a tumor that we decellularize, and we try to measure the mechanobiology now. And that is certainly, it's another great component. Again, this is like the tip of an iceberg. There's a world. And there are also a lot of very good people that operate in this area. It's getting very crowded.
Very interesting talk. I have the question here. Do you have experience with the Matrigel? And how will you compare Matrigel with collagen 1 based gel?
Matrigel, of course, that's how a lot of people worked with Matrigel for lots of time. But the Matrigel doesn't have any real 3D structure. It doesn't have a real porosity. It's not something you can control. Matrigel is a true hydrogel. And so you cannot work on the mechanics. You cannot change really the stiffness. And the other thing is that you don't have a real growth of cells on something that is similar to the extracellular matrix. Matrigel provides great contribution in terms of factors, but not as much in terms of support. So that's why we decided to move to these. Yes.
Yeah. Okay, thanks Ennio for such a wonderful presentation. Very short question. I see in these profiles that some of the things that seems to be pointing out this 3D culture is that you have somehow an epithelial mesenchymal transition signature, actually. So
yes,
that's very interesting because that's what happened when you culture, for instance, cancer stem cells in non-attachment conditions. Probably because of this, and this is related also to the hypoxic conditions, you might have cells which are more aggressive in vivo. So I'm wondering if this has something also to do with the fact that perhaps cells might not be attaching in the same way that they do in to the plate, for instance. So yes, I'm wondering if, for instance, the pathways related to the molecular focal adhesion might be also different. So
they are, and I don't know if I put anything about. No, I didn't. But we looked at that. This is like became like this gigantic paper that it's uncontrollable. But yes, we looked also at the adhesion molecules, and you are right, they completely change. But it's normal because the MDA231, they, especially over time, they become even more migratory. They develop an even stronger migratory phenotype, and so they downregulate adhesion molecules. Again, the problem is that each one of these little areas, it's an ocean per se. And the problem that we're having is that I know that this paper will never see the face of light because reviewers keep asking new pieces, and it's now, we are at 78 panels, the final paper. It's just, yeah, it's kind of crazy, but yes, it's another good point. I gotta go.
We have to move because we are very late on time, and we have the second